My name is Eric Schassberger, and I will be presenting how the lifespan of Artemia is affected by global warming. Today, in today's society, everybody pretty much owns a car. Even myself, I do own a car, and every single year, 1,540,000,000 metric tons of CO2 is released from cars on an annual basis from all the cars that are all around the world. And this does not include the amount of CO2 that is released by any organism that does some type of respiration that releases CO2 as a byproduct, and also any factory or other place that releases CO2 as a byproduct as well. And the reason for introducing CO2 is because CO2 is a greenhouse gas along with methane, and the increasing amount of CO2 in the atmosphere every single year is creating global warming, because CO2 and methane are both greenhouse gases, which lead to global warming. Now, global warming is one of the biggest problems today that are looked at by scientists, and the reason for this is because as the global temperatures go up, it can have devastating effects on different environments. And also another effect of global warming is also a raise in CO2 levels, pH levels in waterways, and also the temperature of the ocean and other bodies of water. Now, some organisms that are affected by global warming, which are much commonly known to the average population, are polar bears who live in the Arctic, who use ice as their hunting grounds. Now, because the global temperatures are increasing, it causes these hunting grounds to decrease as the ice is melting. And then in Antarctica, where the penguins live, they have less room to live because also, again, in the same scenario, the ice is shrinking as well. And then koala bears, their food source is depleting because as the global temperatures go up, it dehydrates the plants that they get all of their water from eucalyptus leaves. Now, one organism that people don't know as well, but is commonly known in the science world, is Artemia selena, also commonly known as the brine shrimp. And these organisms can survive in multiple conditions, and they are also used for multiple kinds of experiments, which leads me to use these as the experiment that I will talk about in a few minutes. These organisms are also very important because they are used as a food source for fishermen or fish farmers who grow certain types of fish that do like to eat these eggs. And also they are the basic building block for multiple food chains in their respective environments. Now these organisms are very important for scientists and for also fish farmers, and I decided to look at how global warming, of all of the effects that I previously said, will affect these organisms. Now, in my experiment, I've used the cysts, as you can see in the top right, all the way till about they got to be juveniles, because the length of my experiment had been 4 days, and over the 4 days, this is about the time that it is said that these organisms will grow to. And the closest studies that I could find to the experiment that I did was a study by Eleanor Boom, who did an experiment where she changed the pH of the water that these organisms were growing in, and by changing the pH, she used different kinds of salts. And when she did this, she found that there was no correlation between the different kinds of salts and the amount of salts that were in each water, in each trial, to how many of these organisms were actually surviving at the end of the trial. And then also another study done by Nana Oposa, she changed the temperature of the water while also introducing garbage, which I did not in my experiment, but she introduced some kind of pollution into her experiment, and she changed the temperature and how much pollution, and if there was pollution or not pollution in her experiment. And as you can see over here, when there was no pollution and the temperature was raised, there was actually a decline in the average survival rate of these organisms. And this led me to my experiment. since I found that there were no studies done that that mirrored the experiment that I chose, I decided to see how global warming would affect Artemis Selene and also organisms that are very similar to them. And the reason for this is because on average 15 million pounds of brine shrimp and other excess materials are harvested each year for fish farmers and for also scientists to use in their experiments, which makes them very important. And for my experiment, I decided to take a 2-day hatching period for these brine shrimp. I put them in a container and I carbonated the water and let them hatch for 2 days, and by the end of the 2 days, almost all of the eggs had hatched besides a few of them who had sunk to the bottom, which were not ready to hatch yet, because if the cysts of these organisms are not in sustainable in a sustainable environment, that they won't hatch and they'll stay in a dormant period. And then after the 2 days after the 2-day hatching period, I took these organisms and I put them into test tubes of 100 milliliters each, and I placed them into a incubator where I changed the temperatures during each and every trial, and I went on a 1 degree Celsius scale. And after the 2-day trial, I took them out, I analyzed how many, I put them up to a white background, and I analyzed them and counted them by hand to see how many of these organisms were still moving around, because typically when these organisms stop or no longer living, they tend to sink to the bottom. And this just shows where what I did with the test tubes when I placed them in the incubator. I laid them all down flat and I let them sit in the incubator for the 2-day period that I already previously mentioned at the set temperature. And this is one of the test tubes after the 2-day trial had been over. And as you can see here, it is a little blurry, but right here is where there is an excess amount of these organisms that are still moving around. And then in the other little circle down there, that is the eggs that never hatched in the beginning period and also never hatched inside the incubator as well. Now, the reason for me conducting my experiment is because there were no sources that were mirroring what I wanted to do. And so this led me to conduct an actual experiment, since I could not find any information through a Google search or by looking at any scholarly articles. So this led me to develop an original experiment where I based most of my work off of different experiments that had already been done, but were not the same as the one I conducted. And for my results, I had found that as the temperature and the pH of the water went up, the hatch rate actually did decline slightly, and then the survival rate of these organisms actually grew every single time, which I generally myself was surprised about, but due to Nana Oposta's study that she had done. But it also is very helpful for scientists that are using these, for people that do use these in their field of work. And as you can see here, I have the control group, the trial, and all the trial groups, and I have the set temperatures and the pH levels for each and every single trial that they experienced, and then on the other side there, how many hatched and how many survived. And this means that since the population increased as the temperature and the pH was changed, this was supposed to model what will happen to most environments as global warming, or water environments, or aquatic environments, in the near future as global warming takes more of a toll on the environment. And it shows that these organisms should prosper in the variables that I conducted in my research. However, because I conducted this in a high school setting, I was very limited to the materials and things that I had to use. So I believe that researchers can use my results to for them to create their own experiments and then see if their results follow the same thing I did. and also I do believe that they should also conduct this on a much larger scale because I only used 4 trials and also a control group, and to make this more of a credible experiment, I recommend using a much larger scale by using more trials and also a bigger sample size within each trial, as I only used 5 test tubes in each trial. And then also, through my experiment, I found that multiple effects on these organisms had little effects on the lifespan and how well they hatched and survived during the 4-day period that I used them for. But based on the experiment that Nana Oposa had done, where the survival rate of these organisms actually decreased, it should be looked into further how multiple effects compared to just single effects of global warming in the future will affect these organisms.
Okay, thank you, Eric.
What were the strategies you used to conduct a review of the literature or gather information? Why did you select these strategies? And which strategies did you consider and reject?
So I have taken research classes in my high school before, and previously I did find that for the topics that I had done previously, it worked out best for me to look at non-scholarly articles and look at the sources that they had used to create their articles, and then take these sources and analyze them and get rid of all the ones that had been non-scholarly and were just some average non-credible person's work, and then also take the credible work that I had found and use this as part of my research, and then use that to develop my experiment.
Okay, thank you. Which of the various perspectives you explored was the most difficult for you to incorporate into your research inquiry, and why?
For my research, I decided to look at the economical, the ethical, and the environmental perspectives of this topic, and the hardest one that I identified was the economic background, because there was little to no information online and on databases of how many eggs every single year, to the exact amount of number, were used and purchased by every single company and that does scientific research, and also fish farmers.
If you had 3 more months to work on this project, what additional research strategies would you put into practice?
If I had 3 more months to research and also conduct my experiment, I would have mostly put most of the time into doing a larger experiment, as I said that other researchers should do if they try to model my experiment, because doing an experiment on such a small scale is, although it does yield results, it is a lot better to get results that are on a much larger scale, because they can be more accurate and more credible.
Thank you.
Thank you.
Thank you.